Good afternoon, everyone. Thank you so much for joining us. My name is Katie Rowley. I'm with the NOAA Central Library as their outreach librarian, and I will be your technical host today. Today, we have our seminar series, the Ecosystem-Based Management and Ecosystem-Based Fisheries Management series. And a few logistics before we get started. If you are an attendee, you are muted, so you can't turn on your mic and speak, but you can put questions in the chat or in the question panel, and I will be asking those questions at the end of the presentation. Or if it's a chat to me about something technical going on with your system, my answer is going to be log off and log back on to GoToWebinar. That solves most of the issues, such as not being able to hear or not being able to see the slides. We are recording today's presentation, so if you do have a question that we don't get to, please know that it was recorded and we will share it with our presenters after the fact for them to answer you offline. Also, if you have to step away for any reason, or if you would like to share this with a colleague, that recording will be up on the library's YouTube channel. And with that, I'm going to turn it over to Peg Brady to introduce our talk today and our presenters.
Thank you, Katie. Thank you so much for your help and your support. Yes, Peg Brady from NOAA Fisheries, welcoming you to our continuing series on ecosystem-based management, ecosystem-based fisheries management for 2022. We began the series a little over 4.5 years ago to increase awareness regarding ecosystem-based management and the principles and highlight the progress that NOAA has made along with many of our partners. And today we have a great example of that. The NOAA Library continues to be a super co-host of the series and posting their archived seminars for us. So it's a great body of information. This is always held on the second Wednesday of each month from 3 to 4 Eastern Time. However, we're going to take a summer break next month, so there will not be a presentation, but we'll resume back up on September 14th with John Hare, who is the Northeast Fishery Science Director. As you know, Katie will take your questions and present those questions to our speakers today. And I want to thank our speakers and our past speakers and all those that have been attending and giving us great advice and counsel about future presentations. So today I'm very pleased to, and we're very fortunate to have Tori Kentner from the Mid-Atlantic Fishery Management Council and Chris Haak from Monmouth University. Great partners in this whole endeavor that was initially supported by the Office of Science and Technology as well as the Office of Habitat Conservation. We also have, as you can see by the front slide here, we have some great partners from the New England Fishery Management Council, New England Fishery Council, and as well as the Mid-Atlantic. And of course, our colleagues up at the Northeast Fishery Science Center. So Tori and Chris, thank you so much for your preparing for today, and I'll turn it over to you two.
Thanks, Peg. Yeah, so this is Tori Kentner speaking. I just want to say thanks for having us today. Chris and I are really excited to share an update on the Northeast Regional Habitat Assessment, which we're calling NRHA for short. Slides. Okay, perfect. So NRHA began 3 years ago with the goal to describe and characterize estuarine, coastal, and offshore fish habitat distribution and abundance and quality in the Northeast. Our steering committee identified 4 actions that were needed to meet this goal. So the first one is an assessment of inshore fish habitat, including fish distribution abundance, as well as associated habitat distribution status and trends. The second one is an assessment of habitat vulnerability, including response to climate change. And third is an offshore assessment, including spatial descriptions of habitat use. And finally, the fourth one is habitat data visualization and support tools. So today, Chris and I are going to share the work that our team has done over the past 3 years to accomplish these goals. So the geographic scope of NRHA covers the Northeast. So it's North Carolina to Maine, both inshore and offshore waters. You can see a list here of our 65 focus species. So these were marked as important to fishery managers. You'll also hear later in our talk that some of our efforts are covering a wider range of fish and invertebrate species that from data we've collected from state and federal agencies. And also we're using those in our community-based models as well. So our products at a glance. So I'll be showing these products later during the talk on the NRHA Data Explorer, which is a Shiny app that hosts NRHA products. It also allows users to explore data trends and species distribution and abundance in federal and state fishery independent surveys. So we started this assessment with a data inventory. Basically, we looked all over the Northeast, looking at different fish surveys, environmental data sets, anything that we thought would be important for running species distribution models, and also understanding fish and invertebrate habitat use. So this research was all stored as a spreadsheet and also turned into metadata documents, which are essentially a one-page overview of each data set that we're using for the NRHA assessment. All of these products you see here, including the metadata pages, are all in the Shiny app, which I'll demo later. So one of our big goals here is that no one has to kind of reproduce all of the work that we've done. It's all captured in the assessment in our products so that we can save a lot of other researchers' time. Our team also created species profiles. So these are a summary of life history and habitat use for each focus species. We're running single and joint species distribution models to help understand habitat use and species distribution. And we're currently writing an inshore report. And then to cover climate vulnerability, our team created a species habitat matrix, as well as written climate vulnerability narrative reports for each focus species. And as I mentioned a few seconds ago, we developed a Shiny app to allow users to interact with NRHA products, as well as some of the fisheries data sets. So we're also working with some of our partners to host some of these products as well. Last but not least, we have several reports and publications in the works, including a community level basis function modeling methods paper and an associated R package. So this slide gives a great visualization of all these products. You can see the data inventory there, the metadata pages, some of the model outputs, some snapshots from the Shiny app, a trawl survey comparison that dives into how each trawl and seine survey, how the data is collected, the NRHA crosswalk, habitat crosswalk, and then a lot of reports. So just to give a little highlight on one aspect that we have focused on is the climate vulnerability assessment crosswalk. So this portion of NRHA integrates outputs from the climate vulnerability assessment, the HCVA, the Northeast Fish and Shellfish Climate Vulnerability Assessment, which is FSCVA, and the Atlantic Coastal Fish Habitat Partnership, ACFHP. So that's a habitat species matrix used in fisheries management. So the major objectives of this was to create a habitat species vulnerability matrix and develop species narratives for 66 managed and foraged species in the region. So basically the matrix identifies the dependence of occurrence of species in specific habitat types while conveying information about species and habitat vulnerabilities to climate change. The species narratives describe kind of the species climate vulnerability, the dependencies and associations across different life stages and climate vulnerabilities of those habitats. So all this information is provided in text as well as in figures, which you can see on the right side of the screen here. So from there, I'm going to pass it on to Chris, who's going to go a deeper dive into our modeling work.
Yeah, thanks, Tori. Hi, everyone. What am I talking about today? I'm going to talk about our models. I guess they've been in construction for quite a while now, and I'm going to try and keep things kind of high-level overview. And if people have specific questions about details of them, please feel free to pass those along. So I'll start with just kind of our basic sort of some of the considerations and that went into what we're doing. Next slide, please. So, what is fish habitat? What defines it? It's a function of a lot of things. The sort of innate physiological tolerances and the performance of animals that determines what sort of physiochemical conditions they can survive in. Also, there are basic ecological requirements. Refuge from predators, is there food available? And that's often a function of other community members. Food is often other prey species and things like that. Life history stage is also a huge component. It's as animals develop their ecological requirements and their performance, their ability to swim, for example, can change drastically, and that can lead to differing patterns of habitat use. And finally, it's a function of dynamic variables that fluctuate and year to year, season to season, and over longer term timescales. So all of these things kind of in our model formulation, we're trying to address in some form or another. Next slide, please. So just a quick review on sort of the idea of community ecology and how communities are structured. So habitat use is a function of again, several concurrent processes we like to think of, in theoretically anyway. So this notion of environmental filtering, that links back to the first thing we mentioned, this idea of physiological tolerances. Are the conditions at a site conducive to an animal, a given species being able to inhabit it, given that species' own limitations? But also going into the mix are biotic interactions. Animals don't live in a void. They bump into each other in space and time and can push and pull each other around in space and time. Predators can affect the distributions of their prey and vice versa. Competitors can exclude each other from space. And finally, dispersal limitations also come into play. Is a given habitat even accessible to an animal given its mobility, or prevailing oceanographic currents, things like that? So all of these things can induce positive or negative correlations in the abundance or presence absence of different species that ultimately lead to the community structure that you end up seeing. Next slide, please. Just a few quick examples of some of these things from the literature. So obviously, the classic idea of competition between rocky intertidal critters. Species that have similar niches can exclude each other from the same habitats if one's a stronger competitor than another. On broader scales, you have things like migratory coupling. For example, right whales and track the migrations of copepods. So the movements of these consumers are driven by the movements of their prey, and these can be pretty broad scale migrations. There's also this idea of non-consumptive effects or fear effects, where simply the fear of being consumed by a predator can alter space used by prey. Likewise, social interactions in species that may share a common predator or share a common prey source, and they might otherwise be competitors, can actually, there's more and more evidence showing that they will often co-occur in high rates, and so that they can exploit information from one another. So in all these processes, well, you tend to think of them happening on fine scales in between individual animals, can have the potential to kind of scale up and impact distributions on broader scales. Next slide, please. So back to kind of our overall strategy here. Based on all this stuff, we've decided to try and take a stage-based approach. So we're partitioning our species of interest into, whenever there's maturity data available, size of maturity data available, we're breaking them down into at least juvenile and adult life stages. Hoping that this will both maybe provide some better resolve any stage-specific differences in stage-specific requirements between, say, adults and juveniles, and or habitat shifts that are going on. We're to deal with the fact that we have all these different animals that do interact and do push each other around in space. We are trying to use a joint species distribution model that sort of allows us to take some of those, that pushing and pulling, hopefully, into account when we're simulating communities, and that will hopefully improve the predictions, and at the same time might give us some insights into what's going on ecologically. And finally, we're making a point of trying to include as many dynamic covariates as possible, rather than static ones, that do reflect the sort of changing nature of these systems, as well as trying to identify predictors that we can really link ecologically directly to animal function. And I'll get a little bit more into that later. Next slide, please. So as most people probably know, SDMs in the species distribution models in the classic sense attempt to just relate observed presence absences or abundances to environmental variables, which are usually, for the most part, physiochemical variables, but can also be aspects of habitat, vegetation, or physical structure, or things like that. And then use that to sort of infer an ecological niche from which you can then go out and project what parts of space and time constitute suitable habitat for species. When you fit a model like that, you end up getting residuals, these over predictions or under predictions that are bound to happen unless you're a god. And those residuals in a single species context just kind of get chalked up to error, sampling error, things like that. Next slide, please. But when you take that in a multi-species context, those residuals actually can provide information. You can look at the residual patterns across species and hopefully garner some information about maybe what's going on between them. So if you have two different species that whose abundances from site to site tend to be positively correlated, that might be indicating that there's something going on there beyond their environmental preferences that's causing those positive correlations. You might have, for example, a predator species that's tracking a prey species. On the other hand, you can see negative correlations, and that might be evidence of competitive exclusion or something like that. But the point is that these, regardless of the cause, these sort of residual covariation, it can be significant, and it can be informative when you're predicting across the community. You can take this residual covariance into account and use it to hopefully generate sort of better predictions and more realistic estimates of species assemblage. Next slide, please. So I'm not going to go too deep into the details, but fortunately there are statisticians who were willing to work with me and who are smarter than me and have helped me to develop an alternative approach for fitting joint species distribution models. As a rule, they're more popular. They're certainly being used more now, but they tend to not scale very well when you have lots of observations. Most of them work in a Bayesian framework and use Monte Carlo sampling that can be really slow. And when you take spatial and temporal issues into account, and when you try and model those things, things slow down even more. And so we kind of came up with an alternative approach to fitting joint species models. It's linked to the probably the most traditional approach, a latent variable model, but instead uses basis functions to get around it. So we have an R package that is actually publicly available now on GitHub, and manuscript is in review at Methods in Ecology and Evolution. And I just want to call out here a big thank you to all those smart statisticians. Next slide, please. So just quickly trying to just cover the basics of how the model works. It's related to GAMs. You know, GAMs describe the relationship between two variables as a linear combination of these things called basis functions, which are kind of almost arbitrary curves, you could say, that are additively combined to describe, say, a broader curve. It's the easiest way to combine it. But so you can also take those. That's in a standard GAM framework. But you can also have 2D basis functions, two-dimensional basis functions, and even take tensor products of them to create 3D things. So you can use that now to describe space and time as well, instead of just a single curve. So basically, CBFM, which we're calling the community level basis function model, uses basis functions to describe covariance between species in space and time, as well as their sort of own within species. And it gives us a fast, flexible way of dealing with that. It allows us to sort of circumvent some of the traditional limitations of assumptions that you make about the separability of space and time in latent variable models, usually, as well as the idea of stationarity, that the fall off in sort of correlation between observations in space doesn't have to, in this model, it doesn't have to be continuous over space. You can have, it's dependent not only on the distances between two observations, but also just the individual locations of those observations. So it's just provides a bit more flexibility. Next slide, please. So we're taking this modeling approach and applying it right now. We've actually added some species, but so it's 100+ species in life stages sampled by the NIMS bottom trawl surveys. These include both the NERA focus species, mostly managed species, but also include taxa that are just appear very commonly in the surveys, and other, that are probably important community constituents, or maybe, and also prey, potential prey species that, based on the literature, for some of these important prey taxa for managed and other common species. So the way the model's set up right now, we're training it based on 15 years of observations from 2000 to 2014, which is around between 9,000 and 10,000 observations, I think. And then we're testing it on the following 5 years. So we're extrapolating up to 5 years down the road to test performance. We're combining the spring and fall surveys when we fit these. Traditionally, I think people tend to break them up separately because there are some issues with, patterns in spring and fall are actually quite different. But using the spatiotemporal structure that we have in our model, it does quite a good job of separating the spring and fall, and so we're combining them all into one thing. Right now we've got 13 predictors in there, and it's set up as a hurdle model with presence absence component and then account component conditional upon presence. We're also comparing those predictions to a zero inflated model, where we model zero inflation coefficients for each of the covariates, as well as their direct effects. And included in here, then we have all these spatiotemporal basis functions and a random year effect to account for year-to-year variability in populations. Next slide, please. So I mentioned that we were trying to find environmental predictors that might be more directly related to animal function. So depth, is a very informative and commonly used predictor for a lot of these species, and for good reason. But there's evidence that when species are tracking, say, changes in temperature, they're altering their uses of depth. And what is it? How do fish measure depth? How do they directly sense the depth that they're at? And how does that affect their ability to perform or function? So thinking about that, we've kind of came up with two other variables that are very closely linked to depth, but maybe have more direct ecological relevance. One is the hydrodynamic environment. How much does the water move in a given location in space? And that has obvious implications for the ability of an animal to swim, to refuge on the seabed, and the costs associated, the energetic costs associated with living in a given place. At the same time, the visual environment is also something that strongly covaries with depth, and also has clear implications for animals' ability to make it around to navigate their environment, to detect predators or locate prey. And so both of those also have, clear morphological, physiological links to the animals exhibit clear physiological and morphological adaptations to deal with these things. The body form of fish, their behavior of fish body forms, their behaviors are very closely related to the hydrodynamic environment they occupy. And likewise, fish and other marine animals have highly adapted ocular systems, visual systems that reflect the visual conditions in the depths they occupy. So we're trying to include these as predictors in our model. So next slide, please. I'm going to quickly just review. So for the hydrodynamic environment, we're using bottom stress as a predictor. It just reflects the intensity of wave-driven water movement at the seabed, as well as the strength of bottom currents. It tends to be inversely related to depth because, for the most part, wave energy for sure, and to some degree currents tend to be stronger at the surface. We're using the 95th quantile metric for this that basically reflects more extreme events, which, based on other work, tends to be a stronger predictor than say median, some measure of central tendency. And these come from the USGS seabed stress and sediment mobility database. Next slide, please. So on the other side, we have these water quality optical parameters, which we're using two different parameters. The intensity of downwelling light, measured as photosynthetically active radiation, and that's basically just how bright it is. And obviously there's a gradient from surface to deeper waters there. And then we're also using this thing called hue angle, which is a continuous measure of the color of downwelling light, and that's influenced mostly by the abundance of it. I mean, it changes with depth in clear water too, because the blues tend to penetrate deeper than other colors, but it's also strongly affected by chlorophyll concentrations and the presence of other dissolved or suspended substances in water columns. So it tends to reflect the sort of coastal to oceanic gradient. Next slide, please. And to try and capture these two things at once, we're using a tensor product of the two. So we get this sort of response surface. So it covers everything from dark to light across the neritic oceanic gradient. And these are the basic characteristics of the water of the underwater optical environment. And they're also light, color, and intensity are where you see the most amount of adaptation in fish vision. And another cool thing about these is they're dynamic. They change with, say, the Gulf Stream position. When it moves closer, you get clearer water in a certain area, or seasonal changes in primary productivity and things like that. So they're quite useful. Next slide, please. So applying all this stuff to the model, we have the out-of-sample predictions are quite good. We compared it to straightforward GAMs as well as spatiotemporal stacked GAMs fit to each species, and the community model outperforms them in pretty much every metric, both discrimination and classification, and it tends to have lower or comparable error. Next slide, please. So just to, what do we get out of this model? Aside from all the usual stuff, so we got smooths for species responses to each of the covariates. We also, for the visual environment, get these plots of sort of these two-dimensional surfaces where we can see essentially the range of optical habitats that they're occupying. So you can see, for example, summer flounder on the left tend to have a broader niche across from the sort of coastal to oceanic zone, which kind of works with the fact that they have this pretty strong seasonal migration from inshore out to deeper waters, whereas the winter flounder are more confined to inshore habitats for the most part. Next slide, please. Another, fundamental thing is just what factors are the most important in driving habitat use for each species. So we do a variance decomposition, and it's a great way of just overall assessing on a species by species basis. And you can also across the community as well, what variables are most important. Next slide, please. Looking at this, these plots when you're looking at it across 97 species get unintelligible. So breaking this down to summer and winter flounder, again, you can see that they tend to respond to similar factors, bottom temperature and annual max surface temp, as well as the optical parameters tend to be among the most influential. But for example, surface temperatures are look to be more important for summer flounder, while salinity looks like it's a stronger driver for winter flounder. Next slide, please. One of the fun things we get out of this is the residual correlation matrix. So this is what we were talking about. These are the patterns in the residuals after you've accounted for species relationships with all your environmental predictors. So these can, this is something that has to be taken with a grain of salt, but it does potentially provide some ecological insights. And it is useful for when you generate joint predictions, your community level predictions. This covariance structure goes back into your predictions. So say when you're simulating a community, if there are two species that are negatively correlated, but the environmental variables suggest they'll both be there, in your simulations, that negative correlation between the two will be evident and theoretically give you some better predictions if you're looking at community metrics like diversity or the number richness or community similarity indices. So these can be potentially evidence of direct evidence of biotic interactions. They might also reflect responses to missing covariates. So if we forgot to include an important predictor in the model, for example, right now we don't have benthic habitat covariates, things like sediment data or structural complexity. If there are species that are responding to those things in a similar way, then we would expect to see maybe a positive correlation between those species as a result of that missing predictor. On the other hand, these can also indicate things like dispersal effects. Next slide, please. So for example, again, breaking this down just to look at a couple of species, you can see there's strong correlations within species, so between the adults and juveniles of summer flounder, for both summer flounder and winter flounder. And that makes sense, both from the perspective of that if we're missing any predictors, we would expect them to show, probably similar responses, but it also could be evidence of dispersal limitations. The presence of juveniles and adults are probably pretty tightly linked as a result of, the juvenile adult connectivity. So there are some other, weaker correlations between the two, between summer and winter flounder overall. And there are you can see there are some other correlations in there that are interesting. Again, they're not to be interpreted as anything ironclad, but they're, maybe provide some insights into what's going on in the system. Next slide, please. So just looking at a couple of predictions. So this is summer flounder. You can see that, for example, the spring and fall distributions are on the left and the right here. So you can, and within the 4 block grids, the ones on the left are the true observations and the ones on the right are the predicted. Sorry, that's confusing. But point being, the model does a great job of, or a reasonably good job of resolving these seasonal migrations that you see in summer flounder. Next slide, please. Likewise, winter flounder, you don't have strong evidence of any seasonal migrations here. But there are some differences in the adult and juvenile distributions, subtle differences. Next slide, please. So what are the next steps with all this? We have to expand our set of predictors to include those benthic habitat variables. That's happening right now. We're working on exploring, looking more at community level metrics to really try and get a handle on how much better the joint predictions are than, say, the single species predictions. We're working on visualizing these results better and finalizing things and make them available to the universe. And longer term, we're working on using these same models to generate long-term projections driven by climate model outputs. And we're looking into the possibility of trying to merge data from some other surveys to fill in gaps in some of the more coastal regions that aren't sampled well by the NIF surveys. And with that, I am going to pass you back to Tori Kentner.
Okay, thanks for unmuting me. So I'm going to go over real quick some of the uses for NRHA. So we didn't just do this for fun. We're hoping that all the products are going to be useful. And so one example already is we've used some of our models for butterfish assessment. So we were asked to provide some of the single species models for the 2022 butterfish stock assessment. So we provided historic distributions as well as projected distributions due to climate change scenarios or under different climate change scenarios, and kind of helped shed light onto some of those links between environmental drivers and stock health and recruitment. We also are planning on using NRHA products for essential fish habitat. So we want to kind of update all of those texts and text descriptions as well as maps. Another one is habitat areas of particular concern. We're hoping to use all of our products and information to update some of those. Also looking for any potential shifts due to climate change and kind of take a more adaptive approach. Another one is the state of the ecosystem reports. So again, NRHA will provide habitat and climate change information on managed species. So we want to make all of our results publicly available. So we're working with a number of different data portals on making that happen. We also have created our own kind of data portal, which is the NRHA Data Explorer, which I'm going to give you a little demo of now. Oh, and before that though, just wanted to kind of overview all of the work that we've done so far. So as you can see from this timeline, a lot of time and work has gone into NRHA. We started in mid 2019 in the summer, kind of identifying our goals, looking at what data was available, then moved into doing a lot of data cleaning, gathering the data, cleaning the data, running models, doing other analyses, and we're kind of nearing the end stages of our products. So summer 2022, we're hoping to finalize and distribute products. Okay, so I'm going to give you guys a tour of the Shiny app. So this is our kind of home for all of our products. So this website is live right now. You can see the URL there is nrha.shinyapps.io/data explorer. So check it out. It's online. We're still a work in progress here, adding new products as they come out, but I would love feedback. So I'm building this website to serve as the home of NRHA products, but it's also a place where you can find fisheries data, especially inshore data. So you can find everything from our model outputs, reports, publication, metadata pages, and then you can also take a deep dive into some of the data sets that we're working with. Really, this kind of started as a way of looking at some of the inshore data sets, and I think one thing that makes this app really unique is that it's the place where you can find all these inshore data sets in one place. So I'll just go through some of these different areas of the website. The survey and species view, that kind of looks at some of the fishery independent survey data in different ways. The model views, that's all of the outputs that Chris just described. And then we have a section for all of our reports and kind of like some metadata summaries and some comparisons of the different surveys. So we'll start on the survey view. So when you land on the survey view, you can choose kind of regional or an inshore view. We'll start with just the NMFS trawl, and these year ranges update automatically. So they'll update depending on which surveys you're selecting. So what you get here is a plot of the top 20 species by abundance. So you can see bay anchovies, longfin squid, butterfish, all of those highly abundant in the NMFS trawl survey. And then you can do the same for biomass. You can also look at a species list. So you can sort this by abundance to see which species are caught the most often, also by biomass. You can search for a particular species. If you're looking at several surveys at the same time, they'll all show up on the species list, and then you'll get an idea of which surveys catch a particular fish. So Acadian redfish shows up in both the main New Hampshire inshore trawl as well as the NMFS trawl. Again, the same analyses that we did before will also populate for whatever surveys you've selected. If you've selected a few different ones, those will populate automatically. Another interesting thing to look at is the cluster analysis. So what this does is it shows you which species are caught together most often. So if two species are caught in the same tow frequently, they'll be closer together on the dendrogram. If they're farther apart, it means they're caught less frequently with the other fish farther apart from that fish. Seasonal temperatures and salinities. So this data comes straight out of the trawl or seine surveys. So we have seasonal temperature and seasonal salinity, both bottom and surface if it's available. So you can kind of look at those for trends over time. If you want to look at an inshore region, you can select that region off the checklist. So let's just look at Long Island Sound real quick for a second. So in the Long Island Sound, we have the Connecticut Long Island Sound Trawl Survey. So again, you get the same analyses as before. You're just looking at one particular region. If you select a region that has a few different surveys, like let's do the Delaware Bay, for example. Again, these year ranges update automatically. These surveys update automatically. And so we can take a look at that. And you kind of get an idea of what species are in each bay or coastal region. Kind of like the regional view, you can select multiple locations at a time. So let's say you're interested in the Delaware Bay and the Delaware coast. You can turn both of those on. This will automatically update to add those surveys, and you can do the same thing. So really the survey page is kind of, if you're interested in a particular area, you want to look at all of the survey data together, this is kind of your first stop. These charts, you can hover over them and you can see the actual values. So that's the abundance in the years, lets you get a little more information out of these charts. There's a few other tools over here. You can zoom, pan, things like that. I think you can even take a snapshot and download the plot as a PNG. So then if you want to take a deeper dive, let's say you're not interested in a particular area or interested in a particular species, we can do the same again. So why don't we take a look at cod? So you can search here or by the scrolling, or you can type into the box. So we'll take a look at Atlantic cod. You get a heat map of the cod distribution. You can turn on and off different seasons. So fall season versus the spring season. You can look at the abundance and biomass across time. Again, this is for the NMFS trawl only right now, but you can turn on other surveys and take a look at a few different surveys, all on the same graph if you want to. You can also down here, you can click on a particular survey if you just want to zoom in on that survey. So it really gives you a chance to dive into all of these federal as well as state surveys at one time. The relative abundance plots. So what the idea behind these is giving you kind of an update on the Elmner Living Marine Resources species fact sheets. If you're familiar with these, you might recognize this. So it's looking at salinity zones, tidal fresh, mixing seawater, as well as different life stages, and then giving you a relative abundance by month. So just using survey data, we're attempting to kind of automate the process. So these are broken down into 20-year bins. So you've got 1960s to 79, 80 to 99, 2000 to 19. You can look at those different 20-year bins. And then you also are kind of seeing the same thing as the Elmner. So you've got your mixing, your seawater. Some of these surveys don't have all of the data, so we're doing our best to kind of fill in what data we do have. but we're breaking it into adult and juvenile, or if there's no stage, we're using no stage, or if there's no salinity zone data available, it's going into the no zone category. So what you see, Maine, New Hampshire, the white area means no data was collected. Lavender means that species, that survey was surveyed in those months, but that species was not found. And then you kind of have a ranging scale from rare, common, abundant, highly abundant in blue, green, yellow, and red. And so you can look at each of those surveys you've selected. This is Acadian redfish, and see where that species was abundant in each month. The last thing you can see for species is a species report. And so we'll go back to Atlantic cod for this. So I mentioned the crosswalk narrative. So that's what these narratives are. So it's a climate vulnerability narrative. So written text, as well as a figure that kind of shows which bottom types are most important, or maybe have the highest climate vulnerability for Atlantic cod. These are still products that we're rolling out. So you might not find all of them in here, but many of them are in here. Let's switch over to SCUP real quick, and we can look at some of the other reports we have. So these are the species profile is a background in life history. So it kind of goes through anything you might want to know about the SCUP or any of the other species we've done so far. A lot of good background information in there. And then essential fish habitat, these are the current designations for EFH for all the species. So next up is our model view. So this is what Chris has been working on. So we put our model results in here. Right now we have summer, winter flounder. We're going to keep building this out as we get more and more outputs, but you can look at some of these correlations. You can look at the distributions for each species. And again, this is kind of work in progress. As we continue to kind of finalize these results, we'll put them in here. For reports, so these are the metadata pages that I've talked about. So each data set that we're working with, we have a different page. So if you're interested in the Delaware bottom trawl survey, for example, you can see who collects the data, what dates they're collecting it, is it available online? If it's not available online, there's usually data access information, so who to talk to get the data, any caveats, methodology overview, a geographic range. So just kind of a snapshot of each data set. One page just to kind of get an idea of what the data is and how you could possibly use it. We're still building this out a little more, but this is our reports section. So we've got our NERA report there. I think we're also working on an inshore report that should be coming out soon. If you want to take a little bit of a deeper dive into some of these different surveys, so this is all the bottom trawl surveys together. So this will give you an idea of what time of year the survey is surveying, the speed, even, head rope, foot rope, survey design. So are they doing fixed or stratified survey areas? What size mesh are they using? So if you want to kind of compare some of these, this is a good stop to kind of check and see how similar are the methods being used by each survey. Same thing for the different seines. And then I also created this survey data timeline. So this shows you, these are a little small, I need to blow this up a little still, but shows you NIMS trawl begins in 1963, and our data goes through 2019, whereas another survey, Maine, New Hampshire, started in 2000 to 2019. So just if you're going to compare these, you want to make sure you're comparing kind of apples to apples. And then I did the same thing for months. So January through December, when is the data collected? So again, so you can compare kind of apples to apples, all surveys that have June, for example, or September, instead of, kind of knowing where your gaps are in your data. Last page is about us. So this is kind of a little overview, what we talked about today, and then lists of all of our partners. Really have to say a special thanks to everybody who supported us, our steering committee, our action team members. All of these people have been, instrumental for this work, and we couldn't have done it without them. So I think that's it. So again, here's our acknowledgements, everyone who's helped us. And then I'll just leave it on this slide so you have our information. As I mentioned, the Shiny app is a work in progress. I'm really building it for you, the audience, researchers, managers, decision makers. So if there's some product or some aspect of the website you'd like to see developed more, let me know. I'm definitely interested in hearing more. Okay, so I think we're ready for questions.
Great. Thank you, Tori and Chris. Okay, the audience, I'm sure, is furiously typing, but I do have a quick question. On the Shiny app, how often is this data updated?
Yeah, that's a great question. So right now we've gone through 2019 and we have plans to update it every 3 to 5 years. And so as you can see by the fact that it's 2022, it's about time for us to kind of go back to these data sources and ask for updates. So hopefully it's going to be a recurring 3 to 5 year process.
Great, thank you. Okay, I'm not seeing any questions yet, so I'm going to ask Peg if she has any follow-up questions she would like to ask.
Yes, thank you, Katie. Appreciate that. Wonderful presentation, Tori and Chris. Thank you so much. Excellent job. Quick question, whether you're getting any inquiries from other regions or even other locations to apply some of these approaches that you're using. I'm just curious whether there's an interest around.
Yeah, I think so.
Sorry, Tori, go ahead.
Yeah, I was just going to say, I think I was on a call with you, Peg, where we were talking to several of the other science centers. I think Alaska and maybe the Northwest also expressed interest in doing something similar in their regions. But hopefully I'm not the one to do it.
Great.
Yeah,
I appreciate that. Thank you.
Okay, we've got some kudos saying great job, really good work. I'm not seeing any other questions, so I'll just ask, is there something you didn't get to tell us that you would like to leave us with? And while you're mulling that over, I do have a question. Could this approach be applied to estuarine environments?
You want to take that, Chris?
Well, first question would be, is that referring to the modeling approach or to doing the, I'm guessing it's modeling. If it is, I will say, yeah, I mean, it's applicable to anywhere that there's reasonably good, reasonably consistent, standardized spatiotemporal sampling, spatially indexed or temporally indexed data. Yeah.
Okay. Yes, that was modeling for sure was the question, but also could the web product be applied to estuarine environments?
Yeah, and I think that is one of our hopes, is to find more data sets in those areas. I think, like Chris said, it kind of comes down to the quality and the quantity of the data available. But as we find more and more data sets, we're definitely going to integrate them into the app.
Great, awesome. We do have someone from Maryland who they said they contributed a couple data sets, and they were wondering if there will be any follow-up with the states about their interpretation of habitat.
Yeah, definitely. We would love to hear from anyone who's contributed their data if they want to have a conversation. We're definitely excited to do that now that we are kind of ending the building phase of this. It's time for us to kind of get feedback from everybody on what they think of the products.
Great, and if there are concerns about results or anything or the survey data, they also would contact you, Tori, or Chris. Who should they contact?
Yeah, they can talk to me for sure. If you're interested in getting any of these data sets, you need to go directly to the data managers. We're not sharing any data, but we are happy to kind of point you in the right direction.
And likewise, yeah, if there's anyone who has thoughts about the modeling work, please feel free to reach out to me, whether it's to tell me that I'm wrong or that you have a good idea, anything like that, please.
Awesome, awesome. Well, I'm not seeing any more questions. You gave us a lot to think about. Oh, there is a question. While that person types in their question, though, I will keep talking. You gave us a lot to think about. Peg noted that we will be taking a break for next month. August is our normal break for this series, but we will be back at it in September. September 14th. If you registered for this seminar, you are automatically registered for the one in September as well. It's getting you all that series. Okay, so I'm looking at the question that just came in. Is there an API plugin to the database backend that hosts these data and model outputs?
We don't have any way, we don't want to share any of the data, and I think our data providers probably don't want us to share it either. So it's kind of just a front end chance for you to explore the data.
Great, thanks for that. Another question. They're searching through the app right now. Perhaps you know, does this include commercial shellfish such as surf clam, scallop, and quahog?
Do have some of that data. It's only from the fishery independent surveys, so no commercial data in here, but there, I think quahog and some of those others are in here. There are also some surveys that specifically target those as fishery independent surveys, which we're working on adding as well.
Great, awesome. Okay, we are closing up on time. Peg, do you have anything else you would like to tell our attendees today?
No, nothing. Thank you very much, Katie, for supporting us here, and also to Tori and Chris. Great job. And to the entire team. This has been an outstanding partnership with fisheries councils, both Mid-Atlantic and New England, and also with Monmouth University, and also with our colleagues at the Northeast Fisheries Science Center. So great job all the way around. Very powerful tools, and hopefully we'll come back and have great examples to share of how this is being utilized. So thanks again to everyone. And, we'll see you back here in September 14th.
Thanks for having us.
Thank you guys for making it run so smoothly.
Thank you all. I hope everyone has a safe and healthy rest of their Wednesday. We did record this, so it will be up on the YouTube channel if you would like to share it. Take care.